Hi, welcome to the Tuesday afternoon live stream at Biology Carol. And today we are talking about monoclonal antibodies and their uses. This is specific to AQA syllabus. So OCRA probably don't need to know all of this content, so you can go off and have a cup of tea. If other exam boards, if you think you need to know about monoclonal antibodies, I do need to say to you that you need to check your specification and you need to check with your teachers and possibly exam papers to see exactly what you need to know and what detail. So this is for AQA, and we're going to be talking about what monoclonal antibodies are, why they are useful, and some of their applications. In particular, with regards to medical diagnoses of cancer, for example, or for other conditions, so pregnancy, and medical treatments, how they can be used in medical treatments. So let's start off with a little bit of context, because this is going to take you back to your immunity topic. Now, hopefully you remember that specific antibodies are produced by specific B cells in response to specific antigens. Now, it might be a bit of a shock to you, but pathogens have many antigens. Oh, howdy to people who are watching. Lovely to see you joining me. So here's the shock, if you haven't realized this already, is that pathogens don't just have one antigen, they have many antigens. And therefore, as part of their immune response, and here's my fine diagram trying to summarize that, is that as a result of a pathogen having many different antigens on its surface, it is going to end up stimulating many different B cells. Notice these B cells have got these antibodies that are specific to these particular antigens on their cell surface. And of course, they undergo cloning or clonal expansion and a bit of differentiation to make those specialized B cells called plasma cells. And it's those plasma cells that make antibodies that are specific to the antigen that stimulated the B cell in the first place. So here we have antibodies with this shape, which is obviously complementary to this antigen here, this one that's complementary to this one, and this antibody that is complementary to this one. When we are infected with a pathogen, we get a polyclonal antibody response because there are bound to be more than one antigens on a pathogen's surface. So now here's the other thing that you need to kind of get your heads around to understand how on earth monoclonal antibodies can be of any use to us. Is that antigens aren't only found on pathogen surfaces. They can actually be any molecule that ends up causing an antibody to be produced. And therefore, that makes antibodies incredibly useful to scientists. But scientists don't want polyclonal antibodies. They want monoclonal antibodies, antibodies of one particular type that will complementary bind or bind as a result of being complementary to one specific molecule. So what are monoclonal antibodies? Here's the definition. These are antibodies that are produced from a single clone of lymphocytes. Notice single is mono, clonal is to do with asexual reproduction of a living thing. Only cells or organisms made up of more than one cell can clone. Proteins can't. That will make sense in a minute. So does it make sense that this, for example, these antibodies here are monoclonal antibodies because they've been produced by one specific B cell or you could say plasma cell. That would be absolutely fine. Monoclonal antibodies can be made by cloning one specific plasma cell so that loads of only one antibody type is produced. We've worked out how to do this, and you don't need to worry about the details of how this happens. Now, here's my little misconception plug, hopefully, well, not a plug for the misconception, hopefully a way of you not having this misconception any longer. Notice that antibodies themselves cannot be cloned. You can't say that a monoclonal antibody is an antibody that has cloned itself to make one type of antibody. And that's because only cells or above are living organisms, and only living organisms can clone. These antibodies are just dear old proteins. They're much smaller than cells, so they cannot clone. So hopefully that makes sense as to why this definition is the way it is, and why it is precise in talking about monoclonal antibodies being produced from a single clone of B cells. Cells can clone. So once we have got these monoclonal antibodies, they are incredibly useful to scientists because they only bind to one thing. And that, of course, is a result of complementary shapes between the antigen binding site on the antibody. So this little bit of the antibody, they are complementary to a particular shaped antigen. Whether that antigenic molecule is attached to a cell membrane or whether it's just free, it's just a molecule hanging around. So what about their applications? Well, here's the absolutely crucial principle, is that monoclonal antibodies can target any molecule as long as it has been able to produce an antibody. So these monoclonal antibodies, they can target any molecule. The molecule, the antigen in effect, the antigen can either be attached to a cell, which means we can identify and target particular cells, or they can be on its own. And that means we can identify and target particular molecules. So first of all, let's look at this scenario first, where the molecule, the antigen, is attached to a cell. And we're going to look at it in the context of cancer cells. Because these, of course, it's really useful to look at it in the context of cancer cells, because cancer cells kind of hide within healthy, normal cells. And yet it's the cancer cells that we want to be able to either detect or treat, and we want to do it specifically. Monoclonal antibodies, incredibly useful here. And they're incredibly useful because cancer cells have different cell markers. They have different shaped proteins on the outside of their cells, which are specific to them. So here we have our collection of cancer cells, and here we have the neighboring normal cells. Notice that their cell markers are different. And this is simply a characteristic of cancer cells. They produce a different cell marker, which is very useful for scientists and medics. So we can use monoclonal antibodies. They can be used to either identify the location of cancer cells in the body by attaching some kind of detectable tag to them. For example, radioactive tag or a fluorescent tag. So here's my little tag attached to the monoclonal antibodies. Notice that the antigen binding site is complementary to the cell marker on the cancer cells, and therefore they bind, bringing about an antigen-antibody complex. And as a result of these nice little fluorescent tags or radioactive tags, we can detect exactly where the cancer is in the body. Oh, I've got a question. Let me answer that. Oh, yes. You what, mate? Thank you. I thought it was only specific to an epitope. Okay, an epitope is an even more specific term for antigen. Proteins can actually have several places where antibodies can bind, and we would call those particular places the epitopes. For the purposes of A-level, you only need to know, for the purposes of getting marks in the exam, an antigen, and it is a more general term for something that binds to an antibody. I hope that helps. You're actually going beyond most A-level syllabuses in understanding and knowing what an epitope is. I hope that helps. Munib, at A-level, do we need to worry about how many MABs were made? The fusion of tumours, oh, how MABs are made, sorry. Right, no, actually, you don't. You don't need to know that detail for AQA students because that was covered back at GCSE. And you wouldn't be expected to know that level of detail at A-level. So I would say, don't worry about that too much. If you happen to remember that it's as a result of a tumor cell being fused with a specific B cell, but specific plasma cell, woohoo, fantastic. Of course, you know, hopefully you can imagine the combination of a tumor cell and a plasma cell brings about loads of plasma cells that produce loads of particular monoclonal antibodies because, of course, they've come from one type of plasma cell. But I hope that helps. Thank you for the compliment, you what, mate. I really appreciate that. Do check out my website, www.biologycarol.co.uk, because that's where all of my videos are, and they all have questions at the end of them. That's me trying to make A-level biology accessible to all students, regardless of how much spare cash is around. And with the effects of this pandemic, I am very, very aware of that. Okay, back to monoclonal antibodies. The other way monoclonal antibodies can be used is by attaching an anti-cancer drug to those antibodies. And that, of course, is going to deliver that anti-cancer drug specifically to cancer cells. And if that happens, it means that that drug doesn't have its effect on normal cells, and therefore side effects are hugely reduced. So here's my little diagram of the monoclonal antibody with the antigen binding site complementary to the cell marker on the cancer cell, and therefore causing the monoclonal antibody to bind to the cancer cell, delivering that drug specifically to the cancer cells. The other classic application, or I should have just gone back, the other classic application in terms of identifying molecules that are free on their own is the pregnancy test. So let's have a look at how a pregnancy test works. And it works fundamentally on the basis that monoclonal antibodies can be made that bind to specifically the hormone human chorionic gonadotropin. Don't worry, don't really need to remember that, in urine. Because when a woman is very newly pregnant, this is the hormone that is produced and it is excreted in the urine. So very shortly after a woman becomes pregnant, this hormone is detectable in the urine, but only detectable by monoclonal antibodies because it's produced potentially in very small amounts. And of course, we need something that is going to bind specifically with that hormone that we're looking for. So here is a diagram showing how the pregnancy test works. Now, AQA students, you don't need to know the specifics of the fact that monoclonal antibodies are bound to blue beads and all the rest of it. But what you do need to understand are the principles by which this pregnancy test works, so that you could explain it if this diagram, for example, were given to you in an exam. So let's have a look. So we have two scenarios here. We've got the scenario where this pregnancy test has been dipped in the urine of a newly pregnant woman. And this pregnancy test has been dipped in the urine of a woman who is not pregnant. So here we have the monoclonal, let's have a look at the pregnant situation first, because this is where the hormone human chorionic gonadotropin is present. And in the pregnancy test itself are a load of free, i.e. they can move, monoclonal antibodies which are specific to that hormone and have a blue bead attached to them. When they bind to the urine, what I should say is when the urine is, when the pregnancy test is dipped in the urine then passes up the absorbent stick that passes through the length of the pregnancy test. And therefore, these monoclonal antibodies specific to the hormone and with a blue bead attached move with the urine up the pregnancy test. When they get to this first window here, they come across a load of immobilized, that means they are stuck to the absorbent, they're stuck to the absorbent stick within the pregnancy test, immobilized monoclonal antibodies that are complementary to another part of the human chorionic gonadotropin. For you what, mate, the human chorionic gonadotropin has two epitopes. Hope that helps. For the rest of you, don't worry about it. The human chorionic gonadotropin simply has two binding sites which will be complementary to two antibodies. These two antibodies here, one of which has the blue bead attached, the other one doesn't. But does it make sense that then a whole complex is formed, sandwiching together the immobilized monoclonal antibody, the hormone, and the blue bead? A blue line appears in the first window. This is kind of saying that the woman is pregnant. But the woman has to wait until the urine passes up to this second window. Because look, there is nearly always, and even in the pregnant woman's case, there will be unbound monoclonal antibodies with the blue bead attached. Notice that in the non-pregnant woman's pregnancy test, all of the monoclonal antibodies with the blue bead attached are unbound, and they are also moving up to this part of the pregnancy test, the second window. And here there is another monoclonal antibody, again immobilized, stuck to the absorbent stick inside the pregnancy test, which binds to the unbound monoclonal antibodies with the blue beads attached. And that causes this blue stripe in this window. The blue stripe in this window tells the woman that the pregnancy test is complete. It's finished. And then she needs to look at in this first window to see if there is a blue stripe there. If there is a blue stripe in this first window, the woman is pregnant. It is about 99.9% effective. There are very, very few false positives. If you get a positive pregnancy test, you are pregnant. I hope that helps, but I hope it helps you understand how the use of monoclonal antibodies has been able to bring about really highly specific diagnostic tests for either particular conditions, you'd call pregnancy a condition, not an illness, or for particular illnesses, cancer, for example. And I hope that has been useful. So if there are any more questions, do start typing them now, because I'm just going to do a very quick synopsis of what we've been talking about. We've been talking about how the definition of monoclonal antibodies are antibodies that have been produced from a single clone or a single B cell or a single plasma cell, those cells that produce the antibodies. We've then gone on to remind you, hopefully, that these monoclonal antibodies can stick to any molecule, or no, they can stick to a specific molecule, and therefore they can be used for certain applications, particularly within medicine. They can be used to diagnose conditions, pregnancy. They can be used to diagnose cancer, for example, diagnose illnesses, cancer, for example, because they can have fluorescent dye to them. Alternatively, these antibodies can also have drugs attached to them, which can then target specific disease cells. The example that I gave you was cancer cells. And the great advantage of using monoclonal antibodies to deliver drugs to particular parts of the body is that they will produce very much fewer side effects, because those drugs will be delivered only to the cells that are unhealthy. I talked about the specifics, the example of a cancer being treated with monoclonal antibodies and with drugs attached. So I hope that is useful. But I think that's us pretty much finished now. So here we are, crumbs, 20 minutes. That's fantastic. Do go off and enjoy the rest of your evening, unless you've got any questions that you want to tap into the chat. Otherwise, I will hopefully see you next Tuesday at 5 o'clock. And it looks like the topic for next Tuesday is the primary defenses that animals and plants have. And that is going to be specifically focused on OCRA syllabus, because that is something that is covered by them. And some of the terminology around that whole topic is particularly confusing. So I'll be hoping to clarify all of that terminology so that you understand that nice and clearly. So hope that's useful. But seeing as we've got no questions, I am going to let you go and enjoy the rest of your evening, and I will see you next Tuesday at 5 to, I suspect, be talking through the primary defenses of animals and plants. See you then. Bye-bye.